Hi, I'm Dr. Jack West, Seattle-based medical oncologist and founder and CEO of GRACE, the global resource for advancing cancer education. Let's continue with our Highlights in Lung Cancer 2011 program, produced by GRACE in partnership with the Longevity Foundation and featuring Dr. Jared Weiss from the University of North Carolina in Chapel Hill. This session will focus on the ALK rearrangement and other newly identified molecular markers and the potential treatments for them.
It would be impossible to do a best of conversation without talking about EML4 ALK. This is actually first starting with a little tour down memory lane, a reminder of how quickly this story has developed. It was only 2007 when EML4 ALK was first reported, and I doubt many of us were talking about it back in 2007. It was really a basic science paper at that point, in Nature magazine, showing this rearranged fusion protein. But I also put this on here to remind you, or to teach for those who were first hearing about this, what a fusion, what a rearranged protein is. Normally, genes have something coming before them that controls whether they're on or they're off. And they should sometimes be on and sometimes be off. One of the things that goes wrong in development, where a cell goes from being normal to being a cancer cell, is that certain things get switched on all the time that shouldn't be on all the time, and other things get switched off. And what happens with EML4 ALK is that you have the ALK, which participates in normal cell signaling, and then the genes are rearranged so that it abnormally sits next to EML4, and EML4 turns it on all the time. And so now you have all that signaling happening that shouldn't normally be happening all the time. Okay, so that fusion gene was just detected in 2007. In my opinion, the biggest happening of 2010 was the data that you're seeing here. Another waterfall plot, I would call it Niagara Falls, showing in the population that has this rearranged gene tremendous benefit from a drug inhibiting it, which we all know is Crizotinib. And the biggest news in my mind of this year was that on September 2nd, 2011, this drug was FDA approved. And so now there is widespread patient access to Crizotinib for patients who have EML4 ALK. Now, if I had this rearrangement, we often call it a mutation, which is fine. My question at this point in this webinar would be, you presented some data on second-line therapy for EGFR mutants. When the day comes that I progress on Crizotinib, what about me? So I'm cheating just a little bit here. At the very end of 2010, Alicia Sequist published her phase 1 data for a heat shock protein 90 inhibitor. Heat shock protein 90 is a client protein. It helps other proteins dock and find their place, and that's particularly important for proteins like ALK. At any rate, what she noticed when she broke down patients by their mutation status is that patients with the ALK rearrangement seemed to derive particular benefit from her heat shock protein 90 inhibitor. And then just this year at ASCO 2011, we saw data from another heat shock protein 90 inhibitor, and again, you can see that the very best results are largely concentrated in the patients with ALK. And so now we have not one, but two drugs targeting EML4 ALK. We have Crizotinib that's FDA approved, and we have a very exciting idea, fairly widely available in clinical trials, the heat shock protein 90 agents. That's actually not it. We also have data for differential efficacy of the chemotherapy drug Pemetrexed, or Alimta. You can see this is the data broken down by mutation status. No known mutation is in blue. Yellow and red are EGFR and KRAS respectively, and the very best results for treatment with Pemetrexed were really in this EML4 ALK group. This data and some other we've seen on this topic have been so convincing to many of us that we've gone back and we have clinically tested our miracle Pemetrexed responders, the people who have been on Pemetrexed for years with good disease control. And I'm not aware of published data on this, but I can tell you by experience and the experience of my friends, we're finding a lot of EML4 ALK patients in there. This is data also presented in 2011 at ASCO from the Lung Cancer Mutation Consortium, showing the breakdown of mutations that they found in their study. And I want to make a few points from this data. First, this no mutation group is now under half. That's pretty huge to me. We're really starting to understand how adenocarcinoma works. During this talk, we've spoken about the importance of EGFR-targeted therapy. We've spoken about EML4 ALK. There's nothing worthy in my mind of best of 2011, but there are ideas about targeting KRAS that we've talked about a bit on Cancer Grace. We've talked about MET. And some of these others do have ideas and trials out there to target them amongst the known mutations. And even since this data was published, we're starting to chip further into this no mutation detected group. On that theme comes ROS1. I first learned about this mutation from another patient on Cancer Grace, and the next stuff that I learned about it was from Dr. West writing about it on Cancer Grace, and it was only very recently that there was actually published literature that I could more formally learn about it. But this rearrangement was actually originally reported also in 2007, just like EML4 ALK. It's actually not technically a mutation, it's also a rearranged fusion protein. So the mechanism is very similar to EML4 ALK. And the test that we see here is also the same. It's called FISH. And a positive test is when the red and the green dots are sometimes separated, which is what you're seeing here. And here's why this is worthy of Best of 2011. This isn't just a new mutation so that we can all pat ourselves on the back and say, hey, we're understanding cancer better. What you're looking at, the left compared to the right, is the first patient formally reported in the literature. And again, I've taken the liberty of cheating just a whiff. This was 3 days into 2012. What you're seeing is a patient treated with Crizotinib, the very same Crizotinib that we've seen to be so effective for EML4 ALK. In this first patient formally reported, you can see quite clearly a dramatic partial response. And the rumor mill says that there are about a dozen other patients so far treated with Crizotinib who have this gene rearrangement, and that results are also very positive. So what about our smokers? We've spoken thus far about all of these mutations, and it's worthy to note that these single driver mutations are much more common in non-smoking lung cancer. Why are we doing so much better at the non-smoking lung cancer than the smoking lung cancer? I promise any of our smoking or formerly smoking patients who are listening to this webinar that it has absolutely nothing to do with a bias on the part of oncologists or investigators. The reason is more biologic. The non-smoking driven cancers are more biologically simple. They have a smaller number of mutations. There tends to be one driving mutation. And so science is at the level where we can start using that understanding to gum the works of the cancer and provide better therapy. Just as a toddler learns to walk before it can run, it's much the same thing with us. Before we're going to be able to inhibit complex genetic changes, we need to first learn how to inhibit simple ones. So I think oncologists are walking. We're doing a very nice job of big advances in these genetically simpler cancers, and I hope as time progresses and we get better that we will one day run and use good targeted therapy for our smoking lung cancer patients as well.
We'll continue with the last part of the presentation by Dr. Weiss in the next podcast.